Well, welcome. Tonight we have a special guest. Going to talk about a topic that people peripherally hear about, but don't really understand. A lot of people in the science world don't understand it. But why don't we start with an introduction?
I'm Dr. Zollie Yavarow. I've got a PhD from Duke University in pharmacology and a master's in bioethics and science policy, and I'm a specialist in gene therapy and rare disease.
Must be fantastic having the bioethics background.
Yes, I love the bioethics background. It's like I was studying sort of mechanisms of disease broadly in the PhD program, and I was thinking more about how we apply the technology that we have in the lab when we're developing, therapeutics. And some of my professors, when I started asking questions, they didn't necessarily know. When I start thinking about, okay, how do we equitably bring this into the clinic? What do patients need? How are we regulating this? Who is going to have access to it? They didn't know, and I'm like, gosh, somebody ought to know. Somebody ought to be thinking about this. And there are people who are thinking about it, and now I'm one of them too. So I've got the bioethics background.
But take a step back. If people would ask you, what do you do?
That's a good question. So I would identify myself, I guess, as a thinker in the gene therapy and rare disease space. So I'm looking to bring different parties together to solve the issues that are arising as we bring this technology into the clinic.
But take a step back and you say gene therapy.
Gotcha. Very good question.
Too many of the political science majors are going to say Levi's. Is that genes? No.
It's not. So gene therapy at its core is using genes as medicine. So genes are coded within our DNA, and there are some disorders where there are mutations in the DNA. So if we think of genes kind of as like chapters in a book, when our cells replicate them, sometimes they make errors. And some of these errors aren't problematic, but some of them can lead to really life-altering disorders. And so if there's something that's broken in gene therapy, we now have the technology to give back a gene that's broken, so we can fix those edits in like a misspelled book chapter.
But many decades ago,
Mm-hmm.
people talked about sickle cell
Mm-hmm.
as being the first target.
Mm-hmm.
That you could have a magic bullet to go in, do some gene altering. And people say, well, how do you do that? You don't take a pill. How does gene therapy actually work?
That's a great question. So we're delivering genes, which is DNA, but how we do that is we actually utilize a virus. So viruses have had, a couple million years of evolution to figure out how to get their DNA into our cells. But what scientists have gone and done is they say, we want to get the DNA that we want into our cells in order to fix something like sickle cell disease. So we're able to put the genes that we want to deliver to our cells as medicine for something like sickle cell.
Viruses, they're like the cockroaches of science.
Yeah.
They seem to have no function in the world except to replicate. Now, you go in and you edit the sequence. How do you target it at the specific area of the DNA to make it work?
Yeah, so for gene therapy, we're not necessarily editing. So within gene therapy, there's sort of other classes. So gene editing is something that we could utilize within gene therapy, but that's not where our technology is currently. So right now, the gene therapies that are approved are basically delivering a gene that is broken. But as we get into editing technology, which is something that's on the horizon, we could have approvals from the FDA for gene editing technologies in humans as early as next year in 2024. And how we do that often is a molecule called CRISPR. So it's basically able to target specific sequences within our DNA.
And how do you control where it snips, where the CRISPR attacks?
Yeah, so we're able to give it a guide sequence. So we're able to say, we want to target, this region of DNA with this sequence, and we want to make sure that that region isn't repeated anywhere else. and that's where it will go. So we have a guide sequence for that.
So in the 3 billion pairs of amino acids, you're taking a sequence
Hmm.
and you're saying, right after this sequence and before this sequence, I want you to take this piece out and put that piece in.
Essentially, yeah.
Now, when you say virus,
Hmm.
I think people will cringe, especially after the pandemic we just went through.
That's true. Yeah.
So how do you tell somebody this is a good virus, not related to the bad virus?
That's a great question. Yeah, so these are viruses that scientists have specifically engineered for this purpose. So viruses have their own DNA, and that's, what makes them dangerous more to us, but we have gone and taken that out. So these are not the same as the viruses that circulate and, give us the common cold.
So you're able to design the virus to carry the load you want and deliver it to a specific place.
Exactly.
Okay, why haven't you designed one that cures cancer?
Some of them do. So there are some gene therapies for cancers that are in development right now. Why we haven't done it for cancer? So cancer is such a big class of conditions. There's not one magic bullet, unfortunately. The other thing is when we're trying to target cells, cancer is so quickly replicating. Something with gene therapy, if we are trying to kill a cancer cell, we'd probably want to get it into all of the cancer cells. And that would be a lot harder to do when you've got something that is replicating. But there are things that are working on.
Well, I mean, realistically, cancer is not an entity. It's a class.
Yeah.
And when you start looking at saying, I'm going to eliminate everything that looks like this, but there's a million different versions of this, you can kill some of them,
Yeah.
but it's not like, I mean, if you could come up with, the ideal, the holy grail of a magic bullet, back in the 80s, people talked about monoclonals,
Mm-hmm.
that we're going to develop monoclonal antibodies to cure cancer. It's going to carry a bomb with it that blows up the cancer cells.
Mm-hmm.
And then they said, all cancer?
It's a little hard to do it for all of it.
So when you talk about gene therapy, it's limited, and we refer to a lot of the indications as orphan conditions, correct?
Yeah, that is correct.
What does that mean?
Yeah, so orphan conditions or rare diseases, as I commonly say, they're defined by the FDA as having less than 200,000 Americans that are impacted by them. But there's actually 7,000 plus different identified rare diseases. So say 30 million Americans, which if you put that in something that's actually like an accessible number, if you pack a crowded elevator, somebody on that elevator is going to have a rare condition.
Well, you got 300 million.
30 million.
Huh?
30 million.
No, but the population of the U.S.,
Oh, exactly.
300 odd million,
Mm-hmm.
and growing every day now, but if there's 30 million, that's, like you say, 1 in every 10.
Exactly.
And the question always comes out, we'll talk about the bioethics later, is how important is it to address orphan diseases?
Yeah, well, I'm somebody who's making a profession on rare diseases, so I might be a little biased. I think it's very important because collectively they're not rare at all. It's like I know multiple people with rare diseases.
Well, I guess I always thought of it as it depends if you're one of the people affected by this rare disease or not.
Yeah.
I mean, you read about orphan drugs and orphan diseases, and it's in the abstract.
Mm-hmm.
If it was your mother, your daughter, your wife, all of a sudden it has a whole different meaning.
Yes. When you get a diagnosis for a rare condition, your life changes for a lot of the parents so a lot of these rare conditions are manifesting and, either newborn screening might pick it up or they've got something that's coming up in those first couple months to couple years of life. Oftentimes it's really hard for them to find a diagnosis, and they get lots of different diagnoses and conflicting things from doctors, sometimes being told, we don't know what's going on, just go home, figure it out. And it's super disheartening for patients, but their lives changed, their lives are upended, so it's incredibly important for a family that has a rare diagnosis.
It's not logical, but you tell me I have a problem, I can deal. Tell me my daughter has a problem. My darling bride has a problem, who's stronger than I am, but it's a whole different world.
Yeah.
And you start looking at it. Do you remember why orphan drug status came about?
So there wasn't a whole lot of interest by the pharmaceutical companies to develop.
Why?
There's so few people, they didn't have the market for it.
It was the exposure. When you start talking about a profitable drug,
Exactly.
the business people in the pharmaceutical companies would look and say, you got to make at least $1 million, $1.5 million a day to make this worth it. Because if there's a problem and we get sued, all the profit for doing this drug could go out the door.
Yeah.
And you sit there and say, it's a shame you thought that way, but if I'm a stockholder in the company, I want my return. So there were a lot of life-altering, I don't know if I'd say life-saving, but some life-saving drugs that just would never see the light of day.
Yeah.
Because the scientists wanted to push it, but the business people said, we're going to make $200-300 million a year on this drug. All it takes is a few setbacks. People die. And all of a sudden now, we're in the hole. So they would keep them on their shelf. Really sad when you thought about how many people could have been saved.
Yeah.
So the orphan drug status basically was some protection from the government.
Yeah, it extended the patents.
Extended the patents, limited, I think it was 7 years. I mean, a patent is usually, 17 years?
I don't know.
And you've taken 10 of those years to move it through development and commercialization. So you got 7 years to make your money back. And when you think about it, today, it depends who you believe, but it's about $2 billion plus to bring a drug to market if you count the ones that failed. Because you start thinking about, why would you bring a gene therapy drug to market when there's only 150, 200,000 people that could benefit?
Yeah, and that's one of the questions that I think the industry is struggling with. So different gene therapy companies have been dropping some of their clinical trials because they're like, this just might not be profitable. And a lot of the funding is actually coming privately. It's coming from patient groups. It's coming from families who fundraise and want to see their children get treated. And I think that's something that's really powerful. It's sort of operating outside of how we might traditionally think drugs are developed. Like, a lot of it is happening with pharmaceutical industries, but I've been really amazed and impressed by the work that patient groups are doing as well to develop gene therapies.
I first ran into it when we were at Unison as a contract manufacturing group. This fellow came in and he was pushing a liposome storage disorder disease.
Okay.
And we know Pompe's, Gaucher's.
Yeah.
He actually scared me because he was so focused on getting this therapy to market. And people who are zealots scare me because they could take shortcuts.
I completely agree with you.
Then I found out, they actually made a movie about him, but we found out that his son had the disease.
Yeah, that is a great film. So the first lab I was in my undergraduate actually was studying Pompe disease. And then in my lab in graduate school, they were also running a clinical trial on Pompe disease. And in my undergrad lab, we watched that movie you're talking about.
And it was, once I understood the motivation, that
Powerful.
back then, 9 to 12 years was the life expectancy.
Mm-hmm.
And he had one child with the disease and one child with early indications.
Something like that, yeah.
John, you started feeling more comfortable with why this guy was so focused.
Yeah.
He left an incredible career at Bristol Myers. We met up with him at University of Oklahoma when he was first starting to promote this. And you sit there and say, okay, this one therapy can save a child for an extra 10 or 20 years.
Mm-hmm.
And it did.
Yeah, yes, that was the development of enzyme replacement therapy, so giving the GAA enzyme that's deficient in Pompe disease.
But now, when you look at products like Cerezyme or, the products that deal with liposome storage disorder, I mean, basically it's a fancy way of saying you can eat all you want, but your body can't convert ADP to ATP, so it can get no energy.
Yeah.
So you were starving, even though you were eating everything in the world.
Mm-hmm.
But let me tap your bioethics.
All right.
$165,000 a year to treat a child.
Mm-hmm.
How do you justify that versus putting $165,000 into the inner city and saving 1,000 children?
Yeah, that is a really tough question, and one that I think the community kind of has to grapple with. Like, I'd say it's an unpopular opinion, but one worth reckoning with. Like, is this the best use of resources? Should we be spending, so let's see, some of these newly approved gene therapies are like over $3 million. Should we have $3 million for one child, or should that be used somewhere else? And that's a really tough question.
I think it's simple. My opinion is going to be jaded by, is it my child?
Exactly.
I hate to say it, but there's no price, there's no sacrifice
Yeah.
that would be too great to save my daughter.
Yeah.
On the other hand, I don't know how to look at the 1,000 other daughters and say, you don't get marginal, just health treatments, just things that could save your life because we're going to save my daughter.
Exactly.
I remember a friend was involved in clinical trials where he was one of the selection board.
Mm-hmm.
And I looked at him and I said, how do you live with yourself? If you really believe that, you really believe that this could save 40% of the children, how do you pick the ones that get placebos? I would be haunted for the rest of my life.
Yeah.
Now he had a very simple answer. He said, you focus on the ones you save.
Yeah. Yeah. And it's a question in the gene therapy field right now, how we're designing clinical trials. Because traditionally, the gold standard is to have a placebo group. But with gene therapy, there's a lot of ways in which that doesn't make sense. So if you have a degenerative condition that's going to progress potentially very badly in, the couple months before you might give the child the non-placebo treatment, is that ethical? Can we do that? Even though it's the gold standard to have a placebo in place. It's what we do statistically.
I don't know how I would deal with that.
Yeah, a lot of parents are choosing not to enroll in that sort of design of clinical trials. And there was a recent decision, Tayshaw Therapeutics just dropped one of their gene therapy candidates because the FDA wanted that placebo double-blind sort of study. And it's just really hard for the industry to do. And it's not necessarily what patients are looking for either, is they want the treatment.
I don't know how I'd make that decision. I got a 50-50 chance for the greater good of sacrificing my child. I can't say that that's okay with me.
Yeah.
You know, when you start thinking about clinical trials, I mean, clinical trials have always been flawed.
Yeah.
I mean, the whole argument about clinical trials are all geared towards men.
Mm-hmm.
Yeah, they are. They were. And somebody would say, why? Is it sexist? Maybe. But I think more than anything, it's, again, the economy.
Mm-hmm.
Men don't get pregnant. You know, so for many
Thought it was
decades,
safer.
it just, you give somebody, I mean, the thalidomide babies.
Mm-hmm.
Nobody ever thought that the children would suffer.
Yeah.
But in the 50s, they got a harsh lesson.
Mm-hmm.
You know, so it was always safer to pick men because they couldn't get pregnant. You weren't
Yeah.
putting infants in the line of fire.
Yeah, and then you don't know what the risk is when it comes to market.
Then half your population is untested. You know, I remember just shaking my head saying, you know, in a way it makes sense.
Mm-hmm.
But again, if you bring it personal, if it's my darling bride or my daughters, I want to know that there's
Yeah.
some testing there that took them into consideration.
Exactly. You want to know, and you want to do everything to make sure they are taken care of.
And you know, we talk about clinical trials. What is a clinical trial?
That's a great question. So clinical trials are what we use to decide if a new drug is both safe and effective. So the FDA is looking for data to answer those questions.
So, phase one,
Mm-hmm.
small group of people, you're testing for,
You're testing for like general safety in phase one.
doesn't matter if it works.
Yeah.
It would be nice,
Yeah.
but you know, the tox work is really, am I going to grow a third arm? Am I going to shut down my kidneys?
Yeah.
It's pure safety in phase one.
Generally, it's actually even more complicated than that with rare disease, though. Times they'll do what's called like a phase 1-2 clinical trial. So they're giving something and they want to know, A, is it safe, but also does it work? So they're trying to do both at the same time. And part of that is because the patient population is so small.
I remember when my darling bride was involved setting up clinical trials for a treatment for broken spine.
Mhm.
And you talk about a rare disease. I hate to call it a disease, but
You can say disorder.
disorder.
Yeah.
You got to find somebody within 4 hours that would typically in a car accident, motorcycle accident, and apply this ointment to it. But you sit there and say, it's hard to write a memo saying there will be no motorcycle accidents except between 8 and 5, Monday and Wednesdays.
8 and 5 p.m. within 100 miles.
So, you know, it was setting up 3 clinical teams.
Mm-hmm.
So you had 24-hour coverage at multiple hospitals, because if each hospital got 1 or 2 patients a week or a patient a month,
Mm-hmm.
you couldn't afford to lose that patient,
Yeah.
both for the efficacy that if your stuff works, which it did, you want to help the patient
Mm-hmm.
because the spinal cord acted like a regular cut. It would scab over.
Mm-hmm.
So if you didn't get it in 4 hours, it was too late.
Yeah.
But you start thinking about what's the motivation for somebody being first in human?
Yeah.
I mean, before you go to clinical trials, you're going to have to do a lot of animal studies.
Mm-hmm.
And that has the ethics and the question of, are you torturing animals?
Yeah.
And that's a hard thing to say. Why would you use animals, Zollie?
Why would you use animals? Part of it's historic. There's definitely a lot of question right now of, do we have better options? Is modeling a better option? Is AI a better option? But historically we've used animals because this was our best way to test before putting it into humans. What's going to happen when we put it into a living being? Is it going to be safe? What might the side effects be?
Well, I could see, you think back way before you were around. When Paul Revere and I had this discussion back then, you start talking about the cosmetic industry.
Yeah.
And how many bunnies were sacrificed until they came up with in vitro ways, fake skin.
Yeah.
And they proved that they could get just as good data out of using fake skin as they could out of using bunnies.
Yeah, and you'd be surprised how many people just want to put different cosmetics on their skin because they want to try it first. But it's different with pharmaceuticals.
And that amazes me because I could see the industry changing over because there was no rational reason to use bunnies. Pregnancy tests. When your mother was around or your grandmother, the old saying, the rabbit died. We're having a baby. I can't imagine.
All sorts of interesting tests.
I can't imagine anybody would do that today.
There might be someone, I don't know.
But you start looking at motivation. The only way I could imagine volunteering for a phase 1 trial is the physician comes up to me, my oncologist comes up to me and says, you got 6 months to live. We've exhausted all known treatments. Well, there's clinical trials going on. Could extend your life.
Yeah. Do you want to try
Could
it?
kill you. Now, if my daughter's about to get married, what do those 6 months mean to me?
Yeah. That's a really important point, is it's a very personal decision. And sometimes 6 months is the difference between seeing your daughter walk down the aisle and not. Sometimes
Seeing her graduate, get her PhD.
Yeah, seeing your graduate get her PhD. Seeing your child go to their first day of kindergarten. It's like those are moments.
That's the only way I can imagine. Now, I mean, some of the trials, like cosmetics, I got a pimple. I'll try some pimple cream. But when I think of chemotherapy, radiation therapy, I mean, that's pretty binary. That's going to work or not.
Yeah, and the question is, it's going to work, but at what cost? What are you going to feel like? How long is it going to give you? Because at some point, something's going to get you. We're all going to die. Are you going to die of cancer? What's your quality of life going to look like after that? Or is you just going to get something else sooner or later?
Yeah, and that whole quality of life, if I could live an extra 9 months but I'm miserable the whole time,
Yeah.
it's easy to deal with it in the abstract. But if you really had to make that decision, it's like, would I ever shoot somebody? It's easy for me to say no. If that person has a knife to my daughter's throat, different opinion.
Yeah.
You know what I mean? It all depends on the circumstance.
Very interesting.
So you go through your phase 1, and it comes out safe. You didn't kill anybody, no 3-headed monsters or...
Yep, no 3-headed monsters, no off-target effects. Safe.
Now you go to the next phase, phase 2.
Yes. We're looking to make sure that it is safe, that it's working in the patients. So often in gene therapy, we're looking to see if it is delivering the gene that we want it to, where we want it to, and to what extent is that gene then producing its product, generally an enzyme.
But you're really looking for efficacy.
Yeah, you're looking for
Does
efficacy.
the stuff work?
Does it work? Does it get where it needs to go? Does it do what it's supposed to do?
So you dose a larger number, but still not a huge number of people.
Correct.
And again, for gene therapy, when we think of some of the clinical trials, I mean, the biggest clinical trial I ever saw was Moderna. They dosed millions of people under the pandemic fast track rule. I hate to say it was a clinical trial, but boy, I felt safe getting my vaccine because millions went before me.
Yeah.
But, if you're looking at 1,000 people, 2,000 people, that's still a good-sized trial.
Yeah, for sure.
And you come out and you say, it helped.
Hopefully.
Well, it
That's
doesn't go
the goal
very
of the
far if it doesn't.
pharmaceutical. Yeah, it doesn't go far after that.
But if it did prove its efficacy, you're done. No.
No, generally you'd then go into phase 3, and sometimes then there's also phase 4. They look
But
for even
phase
more
3,
data.
is it safe to say phase 3 is a large phase 2 that takes demographics into consideration?
Generally speaking, yes. They're looking for phase 2 but greater. Sometimes they're looking more at the dosing information. So something that we do in gene therapy clinical trials is we might be looking at a high dose or a low dose. So with a lower dose, you have fewer side effects, but maybe that's not enough to have the desired effect. Whereas if you have a higher dose, hopefully you're getting more medicine, maybe more is going to the desired area and having an impact, but there also might be a higher immune response.
So you hear about phase 2A, 2B, trying to get the therapeutic range narrowed down.
Yeah.
I mean, you think of things like certain vitamins. They're good for you, but at a certain level, they're toxic.
Water will kill you.
Yeah, the MD50, I remember when I had to do the material safety data sheets, at least that's what they used to call them, now it's just safety data sheets. I learned that the buffer had water, yeah. And if you drank 18 gallons of water at one sitting, 50% of the people will die. 18 gallons?
It's a tough one.
And then I learned that sodium chloride, good old NaCl,
Yep.
if you ingested 6 kilograms, the LD50 was met. How could you possibly eat 12 and a half pounds or 13 pounds of salt?
Luckily, we don't.
So, that therapeutic range sometimes seems silly.
Yeah.
But when you're talking about things as potent as some of the gene therapy procedures, getting it right is kind of important.
Yeah. And we do have a pretty good idea from studies as to what that therapeutic range might be, and that's where we start.
So back to the question of why animals, until they can come up with things like synthetic skin that or pregnancy, monoclonals for pregnancy detection, I don't know that I want my significant other to be taking something that has not been in a mammal.
I overall agree with you. It's like I can see why people are against animal testing, and there have been situations where it's been less than ideal. Broadly speaking, there is so much regulation and so much safety and care about the animal's well-being. I've, worked in laboratories, and we care a lot about those mice or, whatever creatures we're working with. And we're always trying to, I think they call it the 3 R's, reduce, reuse, not recycle in this case. I don't remember what it is. Replace. We want to replace. So when we don't have to use animals, we don't. We'll work in cell systems. We've got organoids now, which is, growing like an organ from cells, or at least an organ-like thing. So we can have
Well, that was the
cells
whole
that beat.
impetus of organ on a chip.
Yeah, it's the basis for organ on a chip, exactly. But yeah, so we put a lot of work into animal welfare, and until we have better solutions, which we're working on, we're looking at AI, we're looking at different computer modeling, organ on a chip, I think it's the best that we can do, and it's important for
And
human
it's still
safety.
tainted by, lack of a better technical term, the fluffiness factor.
Yes.
That people don't tend to worry so much about mice and rats.
Yeah, do you feel different about a rat in your experiment versus a rabbit or a cat?
I got to admit, I do.
Exactly.
I remember when I used to go to USDA in Beltsville, they had the beagles. Schizoid beagles. Because beagles by nature are friendly, they want attention. But when you walk towards them, they'd run away. They'd be barking, calling you for attention. But they knew every time a human walked towards them, they got poked, prodded, stabbed. So they'd all run away.
Yeah.
And you sit there and say, my God, those poor little beagles.
Mm-hmm.
So, again, this is where scientists get a bad rap.
Yeah.
Because a lot of the work we did was to eliminate animal testing.
Exactly.
I mean, most chemists and biochemists aren't the deranged monsters that want to torture little beagles.
No, I cried one of the first times I had to kill a mouse. It's awful. It's awful to do. It's brutal, but it's like I get through it knowing it's like
Mouse
this is
or
for,
my sister, my daughter.
yeah, this is for the benefit of people. This is for the benefit of, the little girl that's in a hospital bed right now. It's like why I killed the mouse. But yeah.
And the security. Now, I've been to Aberdeen Proving Grounds where they worked on neurotoxins, weapons, and where you got assigned a Marine and you couldn't move one step without him watching or her watching you every move.
That is some next level security.
Well, it hits you when you first go there to the guard shack and you get all signed in and everything. The next fence is a big sign that says, use of lethal force is authorized beyond this point. Okay.
Yeah, that definitely hits you a little bit. You're like, all right, I'm listening.
I think this time I will listen to all the orders issued to me.
Mm-hmm.
But I never saw security like when my darling bride was first working at Hazleton. Their animal facility had as tight security as the neurotoxin facility.
Yeah.
Because people thought they were doing good by releasing animals.
Mm-hmm.
And, the sad part, like you say, nobody wants even a mouse.
Yeah.
A mouse is a cute little thing.
Yeah. They
You-
weren't meant to live outside.
So when you say gene therapy, we starting to understand what it means.
Yep.
How do you identify opportunistic areas for research?
That's a great question. So one of the places where our technology is focused right now is on providing genes when that gene is no longer functional. So if we understand a condition really well, so lysosomal storage disorders that you were talking about earlier, it's one gene that gets mutated, it doesn't do its job, essentially. So that's a pretty good candidate for gene therapy because we can give that one gene that is broken that does do its job, and we can correct this pathway that is, going on in there.
Now, is one of the things that works against us the fact that it's a one-shot treatment?
Exactly. So one of the challenging things about gene therapy is that we currently can only give it once. And that's in part due to an immune response. So when our body sees a virus, it wants to attack it. It's what it's supposed to do. And after you've seen a virus, your body has antibodies against it, and it knows next time it sees that. And it's the premise of how our vaccines work. But how that manifests in gene therapy is once we've given it once, your body recognizes that virus. So if you were to give it again, your body would attack it, and it wouldn't work. And it would actually have bad side effects. So we can only give it once right now, and that is an issue for the technology because, what if you have another treatment down the line that might work better? What if it doesn't work as well as you'd like the first time? Say you're in the low dose of a clinical trial and you retroactively wish you could have had a high dose. You can't have it again. And that's something that's really challenging, both from like a decision-making perspective. We want to make sure that patients and their caregivers understand that when they are making decisions about receiving gene therapy, but also from the pharmaceutical perspective, because unfortunately everything does kind of run on money. If the scientists could figure out how to give it again, that would open up a whole new market for gene therapy.
But now you start thinking about that and you say, why don't you just develop another virus?
That's
Have a red virus. I mean, you get infected with a virus.
Mhm.
So they call up to the spleen and they say, we have something down here that doesn't belong. Spleen makes its IgMs and IgGs and sends out the attack force.
Mhm.
but keeps the memory.
Yes.
That once we wipe out this red virus, if it ever shows up again, I don't have to make it. I can just synthesize it from memory.
It's got antibodies.
I got antibodies ready. You know, the IgGs are going to be waiting on it.
Yeah.
But now, why wouldn't I just develop a blue virus to deliver it?
I think that comes back to the resources question we were talking about earlier. So if we're already talking about millions, billions of dollars to
$2
help
billion
treat one
to develop
disease,
the red virus.
do we want to put that same money towards developing another one for the same condition? In theory, we could, but we would then need two viruses that work just as well.
That's fascinating because if you think about it, I'm already dealing with an orphan drug or an orphan condition.
Yeah.
200,000 people. If 10% of the people that got the red virus treatment could benefit from a blue virus treatment, now you're down to 20,000 people.
Yeah.
And yeah, you're right, because you're better off from a financial point of view of putting that money into another indication.
Yeah.
And using the red virus for that too.
Yeah. One of the questions in the field right now is, can we have an expedited sort of approval process for using the same virus to deliver a different medicine, a different gene product?
Well, that would make sense because you think about phase 4 trials. I don't have to repeat my phase 1 because I'm taking the same drug and targeting a different indication, a different disease. So why do I have to repeat the safety end of it?
Exactly.
So wouldn't this be the same kind of idea that if I've shown you that my red virus is acceptable, I'll never say safe because there's always some,
Yeah, there's always something.
but it's acceptable. When the alternative is death, a lot of things are acceptable.
Exactly, yeah. When you're facing death, a lot of things feel better. Yeah, that's one of the questions, like, can we, I think part of the issue is it's kind of a new front. Like, we've not used viruses quite in this way before to deliver genes as medicine. You could argue, like, which part is the medicine and which part is maybe just, like, the vehicle delivery system. So can we use the same virus? We can use a blue virus for multiple different diseases, a red virus for multiple different diseases.
But Zollie, aren't we talking the same questions that we ask about chemotherapy?
In what way?
Well, I think of chemotherapy as having the oncologist balance how quick do I kill the patient versus how quick do I kill the disease. You know, when I think of the effect on stem cells, we need stem cells.
Yeah.
I mean, they are the building block that you hope will differentiate into all the cells of your body. But they're fast replicators.
Yes.
So is in cancer. So if I put something in your body to kill all fast replicating cancer, it's going to take out stem cells too.
Gotcha.
So the question becomes, how do I do the balance? Where do I draw the line of how quick I kill the patient by wiping out their stem cells versus killing the cancer?
Yeah. I think one of the questions you're getting at here is about, like, the dose and sort of how do we give enough gene therapy and does it get divided? Like, where is that balance coming in? And I think what that comes to is, are we incorporating a gene into the genome of the cell, in which case it would be replicated when a stem cell or other cell is dividing, or is that gene not being incorporated into the genome, in which case, say we deliver 4 copies of a gene to a cell, when that cell divides, splitting it, now each cell has 2. Divides again, now each cell only has 1. Divides again, only half of those cells even have a single copy of the gene. And so that's one of the things that we're thinking about in terms of when we might deliver gene therapy. So some gene therapies will target the liver. All of our blood circulates through the liver, and the liver, a lot of the viruses have pretty good specificity there. And the liver grows quite a lot throughout our lives. And if those cells aren't incorporating, if you give this into a child as you grow and your liver grows, suddenly you don't have the same amount of that gene in each cell of the liver as you did before. Will it still be effective?
So the percentage of cells gets diluted as I grow.
So the amount of gene that was in each cell, if it wasn't incorporated into the genome, will get diluted as those cells divide, as the liver, for example, grows.
But now when you say that, do you scare the political science majors that you're going to redo the human being?
Oh, so you're asking about incorporating.
Yes.
Yeah, that's a really good question. That's a kind of a big scary one, I would say, in gene therapy. So when we're incorporating into the genome, we're not changing the human being. What would actually change human beings is if these changes were incorporated into the germline. So in egg and sperm cells, and then those same cells were used to make a child. Yeah. So one of the things that we're really cautious of in clinical trials and everybody's very concerned about is, are there any changes, are there any off-targets effects happening in the germline? Because there's broad consensus among scientists, bioethics, everybody involved in this space that we do not want to be editing the germline. We do not want to be changing.
Well, that's the scary thing where, can I have a designer child? I'd like him or her to be blonde with blue eyes, have this physique. That's a scary movie when you start playing God.
That is a very scary movie. That's Gattaca. Have you seen
Yes.
Gattaca? I watched that one almost every single time my teachers were out in high school, so I watched it a lot.
But you think about it, there's things that Jules Verne talked about.
Mhm.
Light beams, that we now take lasers, little pens that flash. You can hit airliners with green laser.
That's true.
Back when Jules Verne was talking about it, everybody thought it was science fiction. Someday they may be able to beam you up. But, you start looking at, how do you keep it from happening? I mean, the chaos theory says no matter how much you control, there's going to be one of those cats you're herding
Yeah.
gets out of the pen.
Mm-hmm.
So how do you control it?
That's a good question. I think it's hard to control it. So have you heard of CRISPR babies?
No.
So this has been done. There was a scientist in China who...
Was that true, or was that urban legend?
No, that was real. Yeah, there was a scientist in China who edited germline in IVF, and there were 2 pregnancies, twins, and then a second pregnancy, one child, who were born with edits from CRISPR. In theory, the goal was to prevent them from being able to contract HIV, was the goal. It was not a particularly well-designed experiment, but it has been done, and that was, I think, back in 2018, to massive, massive uproar from the community.
Well, when you say it wasn't a well-designed experiment, how could it be? It's not like you're going to go in front of an IRB.
Yeah, exactly. It's not like this got approved. That did not get approved.
I'm going through an institutional review board to say, I'm going to play God.
And that's why we've got institutional review boards, so we don't do things like that.
And I think a lot of people don't understand how well controlled clinical trials are. Because you do have the crazy people like the guy in China.
Yeah.
My Lord, you can't even think that way.
Yeah.
Because who knows what was going to happen to those children as they grew up.
Yeah.
But on the other hand, if my child had a disease that was liposomal disorder, storage disorder disease.
Mm-hmm. Yeah, the lysosomal storage diseases.
The lysosomal, I said liposomal. Pompe's, Gaucher's.
Mm-hmm.
You start saying the odds are very strong that I'm going to get 9-10 years with my baby.
Yeah.
I don't know what I would decide.
Yeah. Getting back to your question of, like, how do we control this? I think my strategy is we control it as best we can with knowledge. So I want people to be educated about gene therapy. That's part of why I'm here. I want people to know that, like, this is a technology that exists. This is something that unfortunately has happened in humans against the vast majority of the field's dislike. How do we make good decisions about this? How do we regulate it? And if people don't know that it exists, if they don't know how it works, and sort of that the technology is out there, how can we possibly keep the cats in the bag, as you say?
Well, you start looking at some of the wild theories, like genetically modified animals shouldn't be eaten. We've been doing gene manipulation by crossbreeding
Yeah,
forever.
yeah,
You start saying, I don't know that a mule was a natural entity. A donkey
Mhm.
and horses and mules. You look at the three of them and say, there's something related there.
Mhm.
The different cows. I was amazed to learn that Monsanto had such a tight grip on the seeds
Yes. Incredibly tight grip on the seeds.
because it's worth a fortune.
Yeah.
But I don't know how you control it because by definition, the plants are going to want to reproduce and they're going to sprout seeds into the wind.
Yeah. Monsanto, I think, has gone and sued farmers because some seeds went to another farmer's field.
Yes.
And they're like, you didn't buy those. And he's like, I didn't try and get them either.
The herbicide that they were using was what they made their money on.
Mhm.
So keeping the line that was resistant to these different herbicides
Yeah.
was worth a fortune. But like you say, the farmer that says, your plants spit their seeds into my yard.
Yeah.
I didn't ask them to, and there's no way I can control the wind. But now you turn around and say in parts of Europe, they make a big deal out of not genetically modified
Mhm.
without really understanding what that means.
Yeah. Yeah. One thing that seems like fear-mongering to me is when people are like, oh, the DNA is like going to incorporate, it's going to get you. It's like,
No.
no, it's not. You eat DNA all the time. DNA isn't if you're eating, you're eating plants and animals, like there's DNA in there. There's DNA in those cells, in your tomato, in your salad, in your
How could there
chicken breast.
not be?
Exactly. You're eating DNA all the time.
But again, a lot of it is the lack of knowledge.
Yeah.
I mean, realistically, some of the ethical questions
Mhm.
that come up have no basis in science.
Yeah.
This idea of the seeds, they're going to modify my body. I don't think so.
Yeah. So with gene therapy, if we're not getting to the germline, we're not changing, the next generation of humans.
And getting to the germline is not a trivial matter.
No. It's not.
I think, when you read about these people that are saying, if I eat a hamburger made from a genetically modified cow, well, how is it genetically modified? Well, they took two different breeds, put them together. It's going to change me. I'm going to grow an extra head or two arms. Back to science fiction.
That's not how our science fiction works in reality. It's like, yeah, I kind of see gene therapy as like Gattaca's coming to life in some ways. We don't have quite that technology. We're not using it that way, but it's like, that's the technology we're talking about. We're not talking about three-headed monsters.
And realistically, when you look at certain disorders,
Mhm.
if you didn't have the science behind what we're doing, those children would live 9 years. They wouldn't survive. I mean, when you start looking at your grandparents,
Mhm.
I mean, my sainted mother lived till she was 94. Not tremendous change to a lot of people.
Mhm.
But 2 generations ago, you weren't living till 94.
No.
And a lot of it, you start looking at rotund people like me who are suffering from diabetes. I mean, right now, you saw before the show, I had a croissant. I know better. The alarm goes off saying you shouldn't have done that,
Shouldn't have had you present.
followed immediately by the phone ringing and somebody from my family saying, we're monitoring you. What did you eat?
We're watching.
But you start looking at the advances in science, and I think more than anything, what you said was profound. Education is the key, because then you can understand how to control what you can control.
Yeah, and what's not going to happen. Like, we don't need to worry about three-headed monsters. We got to fight the right battles.
Where do you see the technology going?
Gosh, I think there's huge potential with it. Like, we're treating in some places, like getting people off of the therapies that they were on before, conditions like hemophilia. It's like, that's incredible. So I think there's immense potential here, especially as we incorporate the gene editing technology as well. But with that, we also have to be incredibly careful about how we're doing it, in what speed we're doing it, and how we're regulating it. And I want society to be making decisions. I want society, like the general public, whoever's listening to us today, to be informed, to be involved in those decisions. I don't want it to just be the people who are, the high-ups in pharma, who are high-ups at the FDA.
No, because again, if you had left it up to, and it's their job to watch out for their stockholders' equity.
Yeah.
But orphan drugs would have never made it out of the libraries.
Yeah.
It's just not profitable enough. There's too much financial risk. Until you look and say, but it's my best friend's daughter.
Yeah.
It's so-and-so's father, so-and-so's wife. And then somehow that $1 million, which is hard to justify, becomes justifiable.
Yeah.
How do people get to learn? If they want to learn about gene therapy, where would they go?
That's a great question. So there's a couple good resources. So the American Society for Gene and Cell Therapy, they've got some excellent resources on their website and have put things together. I just founded my own organization where I'm trying to educate, providing seminars at libraries. We've got a public library or other source that would like me to come and share about gene therapy. It's called Zaris Consulting. So I'm interested in just helping people navigate these complex decisions and information that's surrounding gene therapy. A lot of pharmaceutical companies, interestingly, are also putting together educational resources. So Vertex has put together, I think they call it Gene Possibilities. So that is one option as well.
Well, thank you for shedding some light. As we come to the end of the show, I want to thank you for coming on.
Thank you for having
me. And as you expand what you're doing, we'll welcome you back.
Yeah, and the field of gene therapy will be expanding too.
And to our 6 loyal viewers, thank you for joining us. I hope you learned something. As always, may God bless. May tomorrow be a better day than today. Thank you.
Not too long since I've been home.